Bismillahirrahmanirrahim. In this video, we are going to study the matrix glycoprotein that are also termed as matricellular protein. Two important protein in this group are laminin and fibronectin. Matrix glycoprotein or in matricellular protein are large, weighing about 150,000 to 1 million kilodalton, and they are multimeric and multi-domain macromolecule with long arms that bind with other matrix molecule and support or modulate cellular attachment. They organize tissue topography, they support cell migration, they are responsible for orientation of the cell, and they induce cellular behavior. So important members of this group are key basement membrane protein laminin that we have also seen in detail in the basement membrane video, and the key stromal connective tissue protein is fibronectin. The laminin is a versatile basement membrane glycoprotein, as we have seen in the video of basement membrane, and it has got a cross-like structure. It is composed of heterotriemers that is proteolytically processed. They self-polymerize into sheets. The laminin associate with type 4 collagen and other basement membrane molecule to initiate basement membrane assembly. They provide a scaffold for basement membrane structure, support cell adhesion, and influence tissue morphology and function. Their binding partners are heparan sulfate proteoglycans, integrins, dystroglycan, and Lutheran blood group receptors. There are many types of alpha, beta, and gamma chains of these laminin that gives them opportunity to form various isoforms. So these isoforms are tissue specific and they are architecturally unique. So the clinical relevance of this laminin, if they are mutated, they will result in muscular dystrophy, and epidermolysis bullosa result from mutation in laminin, integrin, or collagen 7 or 17. So the role of laminin in the healing of the wound is that the proper processing of epidermal laminin isoform is critical for the reepithelialization, and laminin binds epithelial integrin alpha 6 beta 4, stabilizing epidermal integrity via interaction with the collagen 17 and collagen 7. This is the illustration that is showing the interaction between the integrin and laminin in the plasma membrane and the extracellular matrix. It is a crucial communication bridge between cell and their environment. The plasma membrane over here, this plasma membrane is showing with these integrin receptors, and these integrin receptors are binding with underlying laminin. Uh, these laminin linking the integrin or the cell via the integrin to the extracellular matrix. These linkers can often be fibronectin, velcolin-like domain, etc., that facilitate the stable adhesion and signal transduction. The down there, these wavy lines are of collagen, and the nodes, these circular areas, are binding domains or anchoring points. So this provides structural and biochemical cues to the cells. So they are the integrins are bidirectional transducer, relaying signals from the extracellular milieu to the cell outside in signaling, and from the cell to the extracellular matrix or milieu. So that is inside out signaling. They are responsible for both, and this connection is essential for cell adhesion, migration, and tissue repair. So further to emphasize that this cross-like structure of the laminin, that is composed of alpha, beta, and gamma chains, specific for organ. Fibronectin is a matricellular adhesive glycoprotein, and it is a versatile adhesive glycoprotein that is found in the stromal connective tissue and wound provisional matrix. It is a V-shaped homo or hetero dimer that is linked by two C-terminal disulfide bonds. Its interaction are with bacteria, collagen, heparin, fibrin, fibrinogen, and integrin. So its major function are to link matrix component and cell, and to facilitate the cell migration, particularly in thrombi and wounds. Its interaction with the integrin is defined the integrin receptor family through its binding specificity. It forms insoluble cellular form and soluble plasma form. There are two types of this fibronectin: plasma and tissue type. And there are about 24 isoforms that are produced by alternative splicing. A point to remember that both of these, the insoluble and soluble types of the fibronectin, are encoded by a single gene. The plasma fibronectin binds to fibrin and promotes platelet adhesion, and stabilizes the thrombi by factor 13 transglutaminases. It promotes keratinocyte migration and attachment in wound healing, and aids in repithelization by interacting with the collagen. It is produced by, especially the tissue type, is produced by the fibroblast and assembled in insoluble fibril with integrin and collagen assistance. It is localized in granulation tissue, loose connective tissue, and provisional wound matrix. So, experimental knockout of plasma fibronectin doesn't alter healing. So, there is compensation by the cellular fibronectin. Thank you for watching this video. If you have any question, please write in the comment box.